Today, I want to talk about DNA indexing with k-mers. We go over the pipeline of variant discovery, and we explain how we could create a DNA index with k-mers and storing them as either a sorted array or hash table. Every human cell contains a set of genetic instructions to maintain cell functions, which is stored as DNA. A polymer or macromolecule consists of four types of molecules: adenine, guanine, thymine, or cytosine. The Human Genome Project was a pioneering effort to find the DNA, to understand what are the molecules that make a DNA. So the human genome is consists of more than 3 billions of these molecules or bases, which gives us the reference genome we use for many types of analysis. So this is very useful because humans share more than 99% of their genome. Here I'm showing three individuals and their DNA, the sequences of uh, these four types of molecules, and we can see that most of them are quite similar, and only this position is different across these three individuals. This is very useful information because by leveraging genomes, we could study different genetic diseases. Also, it is useful for cancer. An important hallmark of cancer is the accumulation of variations and mutations. So there are different studies over years that they benefit from genetic analysis to study different genetic diseases. So in this type of analysis, we first extract the DNA from a cell and we put it in a sequencing machine. So we put our genome or DNA to this machine and it gives us some DNA sequencing reads. The challenge is that the machine cannot give us the whole chromosome or the whole DNA. And it gives us some chunks of DNA, maybe a few hundred bases or more. Recent technology can give us a few thousand bases. So we use this information, and remember, we have the reference genome. So we could leverage both and try to find the variations. So we have this sequencing machine, and we sequence the DNA of my sample, and we have these chunks of DNA. So during a process called read alignment, we try to map each of these reads to the reference genome. For example, this read maps to this position. So we can see this is our sequencing read, and the top one is the reference genome. And we can see it's quite similar, only the difference is this position, that instead of the molecule A, we have a C. We could continue for other reads, and we can see we could map all of them back to the reference. And these positions that we have some differences is important for us. We want to find these differences, which are called genetic variations. It's important to notice that there might be some sequencing error. The machine can give us some errors. So we need to distinguish the actual variations and the sequencing errors to find the true variations. We are interested in today's lecture on this part, read alignment. So we have those sequencing reads, and we have the reference genomes here as a simple example of as a book that we want to find all the places where the specific string occurs in. The example here is GATACCA or Gattaca. We want to know where this string occurs in the reference. Let's put it more formally. We want to align a sequencing read to a genome. We have our reference genome or our text as T, and it has like 3 billion characters. And we have a pattern, our sequencing read, with around 100 characters. And we want to find the pattern in the text. Our string is a with the regular definition is a finite sequence of characters, and our characters are drawn from alphabet of A, C, G, and T. And the alignment is finding the coordinate of P in the text T. So one naive idea for finding the exact matches of our text P with the length of n onto our pattern P with the length of n onto the text T with the length of m is just slide this pattern, slide this over the text. So we could start from here and move it and slide it all to the end. We could compare each of the characters when we are sliding. So the question here is that how many character comparison needed in the worst case scenario? So we have our long text tm. So at most there could be m minus n plus 1 sliding windows. And every time we compare n characters. So in total, we need this amount, m minus n plus 1 times n number of comparisons. It's quite a lot. We could benefit from the idea of book indexing. So in many of books, if we go to the back of the book, we can see a kind of index. It it has all of the key terms, and they are ordered alphabetically. So when we want to know, for example, the pigeonhole principle, we could check the index and find the page number and go to that specific page to study pigeonhole principle. This is useful idea and analogy we could benefit in our problem for DNA sequencing alignment and finding the matches. So we need to index our DNA. We have our reference genome or our text T. I put an example here, ACGT, ACGGA, and we want to index it using k-mers. So the challenge is that why do we need to use the idea of k-mer? Because there's no specific distinction or idea of word in the DNA. In the book, we have different words, and we could put them and index them using those. But in DNA, we need to define some type of words. So here, the idea is to use k-mers. These are fixed size, or we call them n-gram. These are fixed size string, a substring of length k. For example, it could be k3. Starting from the beginning, we want to extract all of the k-mers, and all of them are trimers. So we have ACG here, and we also store the offset, the starting position. So for ACG, we put the k-mer here and the offset 0. If we go one step ahead, the k-mer is CGT. We put it here, and the offset is 1. Next, the k-mer is GTA at offset 2. Next one, TAC at position 3. Remember, we want to make an index out of it. So we need to keep the k-mers sorted. Up to now, these are sorted alphabetically. We have A, C, G, T. If we go to the next k-mer, ACG, to keep the k-mers, our index sorted, we need to put it here at this row. So we could move the rest a bit, shift them, and put the ACG here with offset 4 at this position. Next one is CGG. We put it here based on the order and with offset 5. And the last one is GGA. This is GGA at offset 6. So we stored all of the k-mers in our index. Now we have a pattern, CGGA, and we want to know whether this pattern occurs in the text T or not. We want to query the index to know whether it's there or not. One idea is to benefit from binary search. In other words, we want to find the location of the pattern CGGA in the text T. So we could focus first in the first part of the pattern, the k-mer CGG. We want to find the location of CGG in T by querying the index. To do so, we compare our k-mer CGG with the middle item in the index of T. So the middle k-mer CGG T in the index, and we compared with the CGG. We compared whether it's like we should go continue searching on the top part or the bottom part. Every time by binary search, we make the search space half. So we can see that CGT is greater than CGG based on the alphabetic order. So this tells us we could ignore the bottom part and focus on the top part. Again, we compare our K-mer CGG with the middle of the rest of the index, which is ACG. So we compare with the middle part, ACG with CGG, and we can see that ACG is smaller. So we could focus on the bottom part of the index, this part, which is the only one is left. So then we could easily compare the last one, CGG, with the our K-mer, which is the same as CGG that we are looking for. So yes, we find the K-mer in our index. This gives us the offset of the text T. So we find the position of K-mer CGG in the index. So we could go over to the text and see at the position 5, at the offset 5, we can see our K-mer. And now the question is whether the whole pattern occurs in the text T or not. We could extend it slightly. We could extend it by 1 and make sure that the whole pattern CGA occurs in the text T. And yeah, we can see that we can do the verification and it's there. Occurs in T at offset 3. And another idea is to benefit from hash table to index our DNA using K-mers. So a hash table here, we could consider it as some buckets. In this example, our table size is 11. We have 11 buckets. And we want to store our maps that are K-mers and offsets. We want to store them in different buckets. So how could we do that? We need to have some idea about the location, in which bucket we want to store different K-mers. Hash functions would do that for us. Hash function, using hash function, we could map K-mers to buckets. We have different K-mers, and to define this function, we could encode our bases, nucleotides A, C, G, and T. Here we are considering like AS1, CS2, GS3, and TS4. As an example of our encoding, GTA can be encoded to 341, just simply putting some as a decimal number, putting G as 3, T as 4, and A as 1, it will be like 341. Another example, ACG, A is 1, so 1, 2, 3, which will be like 123. This number is quite big, but our bucket size is 11. To make it smaller, to make it fit in the bucket, we could use a kind of mod function as our hash function. So the input to the hash function is our K-mer, and we could encode them using this type of encoding. And by calculating the remainder of division by 11, it gives us a number, which would be smaller than 11. There are different types of hash functions. This is just an example. So we have our hash table, which is empty for now, and we have text T, ACGT, ACGG, and we have our hash function here. And we want to go over text T and extract the K-mers one by one and put them in the buckets. So from the beginning, we have ACG at offset 0. So offset 0 is ACG. We could calculate the hash function. Encoding of ACG would be 123. Mod 11 is 2. 2 tells us the bucket ID. So we want to put our K-mer offset ACG and 0 in the bucket 2, which is this bucket. So we can put it here, ACG and 0. So it's kind of linked list that we store k-mer and offset map in the hash table. Next, k-mer is at offset 1 is CGT, which we can calculate hash function as 234 mod 11 would be 3. 3 tells us that it's a third bucket. Here we are starting, our bucket ID starts from 0. So this will be the third bucket, and we put CGT and 1 here. Next one, at offset 2, k-mer is GTA, and we could calculate the encoding 341 mod 11 would be 0, which goes here. So we could put GTA and offset 2 in this bucket. Next one, k-mer TAC at offset 3 would be 412 at, by calculating the mod 11, it gives us 5. So bucket 5. We could store TAC3 there. Next one, offset 4 is ACG. And the encoding is 123, which gives us the bucket 2. Oh, it's quite, we already have similar one. So what could we do? We could just simply first find the bucket. Bucket 2 is here, it's full. But we could consider as a linked list and continue putting our k-mers one after each other. So we could put ACG at offset 4 here. Just right after, it's a bucket, we could put a different stuff in it, but we need to have this kind of linked list to know how to, then we want to query the index, we need to find those. So we have ACG for here. And lastly, our k-mer CGG, that's offset 5. Also, it's different k-mer, but our hash function gives us the same bucket. Bucket 2, so we could put the last one here. This is called a collision. Then the hash function, the hash value of these two k-mers are the same. So we have our hash table. Now we want to search or query the pattern TAC. We want to know whether the pattern TAC occurs in the text or not. To do so, we calculate the hash function of this pattern of this k-mer TAC, and it gives us the bucket 5. So we go to bucket 5, and we do we compare the k-mer with all the stuff that is in the bucket one by one. Here is only one k-mer is there, so we could see that, oh, it's actually TAC. So the k-mer is the same. So we could find out that the pattern occurs in T, and this map tells us it occurs at offset 3. It's quite, it's very fast. Hash tables are very fast. In summary, we discussed how to index a k-mer, which is a straightforward way, and it can significantly reduces the time cost on alignment. We could also discuss how to query the DNA index. You have two ways: the sorted list and the hash table. That's all for today.